Propofol is an intravenous anesthetic that can directly activate and potentiate GABAA receptors. Despite its widespread use in clinical setting, many aspects of its mechanism of action are not well understood. The goal of the present study was to establish the functional role of amino acid residues in the putative propofol binding site and the GABAA receptor. We took advantage of a recent photolabeling study that had placed the propofol binding site near the extracellular end of transmembrane domains at the inner subunit interface. By mutating sites near histidine 267, a residue photolabeled by ortho-propofol diazirine, we identified several positions where an amino acid substitution affected receptor activation by propofol. Specifically, tryptophan substitutions of tyrosine 143, phenylalanine 221, and glutamate 224 reduced or eliminated activation by propofol, without affecting activation by pentobarbital, which is another allosteric activator. Interestingly, we found that the mutations did not affect propofol activation in GABAA receptors consisting of alpha-1 and beta-3 subunits. One explanation to this is that unlike the beta-3 homomeric receptor, in which mutations modify all inner subunit interfaces, the heteromeric alpha-beta receptor retains some unmodified interfaces. But much to our surprise, we found that combining a beta subunit mutation with a homologous mutation in alpha still did not affect activation by propofol. This finding led us to consider the existence of non-equivalent binding sites at different inner subunit interfaces. A possible alternative interaction site is near methionine 286. This residue is located in the beta-alpha interface and is photolabeled by meta-azipropofol. To test the role of this additional site, we created receptors in which a tryptophan substitution at methionine 286 was combined with tryptophan substitutions at positions 143, 221, or 224. In this way, mutations were introduced to each of the 5 interfaces in the alpha-beta receptor. We found that the combination of mutations led to elimination of receptor activation by propofol. We conclude that propofol can interact with any inner subunit interface in the alpha-1 beta-3 GABAA receptor. Accordingly, all interfaces must be mutated to abolish activation by propofol. Our data also show that the binding sites at different interfaces are not identical. We propose that at beta-beta and alpha-beta interfaces, propofol interacts with a site near histidine 267. At the beta-alpha interface, however, propofol binds near the methionine 286 residue.